Hi there. In this video, we'll discuss stem cell technology and explore its potential applications for tissue engineering and treatment of disease. In this video, we'll talk about the promise of pluripotent cells, why everybody is so excited about them. We'll talk about how you define various stem cells, and we'll spend a fair bit of time contrasting embryonic stem cells with adult stem cells, highlighting their advantages and disadvantages for various uses. One of the huge exciting potentials of stem cells is the ability to turn into any of the adult tissue types from any of the three germ layers, the ectoderm, the mesoderm, or the endoderm, or in addition, producing or the germ cells themselves. These cells in the inner cell mass at day 5 eventually differentiate into these three layers, eventually giving rise to an entire organism. Now, there is a book from 2001 that is quite comprehensive that will give you lots of information on this link right here. I've also on the home web page linked to a much more concise fact sheet and a little bit more up to date on stem cells, but both of these would be excellent reading for this material. The second promise of stem cells, which is related, is not just to make any tissue de novo, but to repair damaged tissue. Here we see the cross section of a mouse heart. What we can see over here is healthy, thick, muscular ventricle wall. Here we see a region that has suffered a heart attack, and as you can see, the muscle tissue is gone. It's very thin. In these two blow-ups of this region, they have introduced cells which they have genetically modified, and those are labeled blue. And then using a different staining technique, they can show that these cells have roughly in the same physical locations developed characteristics which are identical to those of cardiac myocytes, the muscle cells that help produce contraction. So the thought is that by introducing stem cells to diseased tissue, you might be able to repair it. And this, of course, relies on the ability of the stem cell to differentiate into cardiac myocytes. Now, a little bit more detail on where the first embryonic stem cells came from, and that was from fertilized eggs in vitro fertilization clinics that were no longer being pursued, either because the people who created them were successful in achieving a pregnancy or no longer wish to attempt it, which are frozen. They have many frozen embryos there, which have at around day 5 this inner cell mass. This inner cell mass, is removed. They're plated out on specialized petri dishes, which have feeder layer cells underneath, and you can propagate pluripotent stem cells from this tissue source. And on this slide, we see a scanning tunneling image, which is not showing too much new information, but just to see, here's the inner cell mass. It's a false color image, but I think it gives you a better feel for what this might look like. And then just to review normal human fertilization, at the time of ovulation, the ovary releases an egg, an oocyte. Sperm have traveled up the fallopian tube, will fertilize the egg somewhere in the fallopian tube. The fertilized egg will then make its way down the fallopian tubes, and around day 6, 7, 8, 9 will implant on the uterine wall if successfully fertilized. So at around day 5, before implantation, you have this blastocyst stage and the inner cell mass, and this is the time period where you can isolate these pluripotent cells. But before we focus on the inner cell mass, let's focus on a completely developing fertilized egg. And here we show a fertilized egg and the various stages of division. And you see the inner cell mass will give rise to the organism. But the fertilized egg is also capable of giving rise to the structures that will form the placenta, so the tissues that support the developing organism. And that is a difference between pluripotent embryonic stem cells. Embryonic stem cells are pluripotent. They can make any single of these tissues. And another term called totipotent, which is what a fertilized egg is, and that can give rise to not only any of the tissues or all of the tissues, but also all the tissues such as the placenta, which are required for the development of an organism. So fertilized eggs are totipotent and embryonic stem cells are pluripotent. And if we're going to define an embryonic stem cell, here are 6 features of it. They are cells that come from the inner cell mass of the blastocysts. They are capable of unlimited symmetrical divisions, regenerating embryonic stem cells, long-term self-renewal, and if done properly, they will not differentiate into any of the terminal cell types. They are, in a sense, immortal. They are characterized as having normal, stable, full diploid karyotypes. This is to contrast it with immortalized cancer cells, which have mutated atypical karyotypes. Their functional definition is that they're pluripotent. They can give rise to any of the cell types coming from the endoderm, mesoderm, or ectoderm. They're also capable of creating germ cells in a new generation, and that they are clonally related, meaning that they have the same genomes. In addition to this self-renewal, which we had talked about in the previous slide, where stem cells are capable of dividing indefinitely, regenerating themselves, they also have this potential for a division called an asymmetric cell division. And it's called asymmetric because the 2 cells which divide are going to be genetically identical, but they will become phenotypically different. And the stem cell can divide into 2 cells, one of them which would renew a stem cell, and the other one would become a terminally differentiated cell, meaning a neuron or an epithelial cell or a fibroblast or a myocyte or any of the other tissues or cell types of which you're familiar with. And as far as I think anyone has known, these cells are capable of unlimited cell division. Now this slide is fairly wordy, but I wanted to have it all on one page where we looked at the different types of stem cells, create definitions so that you can help keep that straight. We've been talking about, so far, embryonic stem cells. There are also types of stem cells called adult stem cells. These are tissue-specific stem cells, for instance, lung or brain, neuron. They come not only from adults, but also children and fetuses. They are multipotent, but not pluripotent. They are committed, in a sense, to becoming a cell from the tissue of origin, which is to say neural stem cells can turn into all the cells of the brain, but for instance couldn't form non-brain tissues. And a major difference with embryonic stem cells is they do not have unlimited replicative potential. They can divide many, many times. They can turn into lots of different cell types, but we're not able to grow them indefinitely in the laboratory, and they may or may not have limited replicative potential in the body in which they're somehow replenished. A third type of embryonic-like stem cell is an iPS cell. These are called induced pluripotent stem cells. And this is the Nobel Prize-winning research from just a few years ago, in which an adult cell, in the cases, I think either fibroblast or epithelial cells, were genetically modified, and they revert to a pluripotent state like an embryonic stem cell. And they have the characteristics of embryonic stem cells in that they're able to form any tissue type, and they have unlimited replicative potential, but they are genetically different from embryonic stem cells. We've modified them, and they may have some slightly different properties. These are questions that are still being answered. Lastly, we'll talk about a somewhat controversial type of stem cell, cancer stem cells. It's thought that Lastly, we'll talk about a somewhat controversial type of stem cell, maybe a cancer stem cell, because of the similarities between embryonic stem cells and cancers, that is, unlimited replicative potential and ability to form lots of different tissues. People long speculated about the possibility that stem cells give rise to cancer, and it is generally accepted in the literature and by most cancer researchers. And this is a subpopulation of cancer cells that can self-renew, can create all the cells that are required in the cancer. And for some types of tumors, like teratomas, can produce many, many different cell types. The idea is that some of the issues related to treating cancers, like chemotherapy, the chemotherapy might be effective against the 99.99% of cells that are not cancer stem cells, but are in a sense a differentiated kind of cancer cell, but that the chemotherapies are ineffective at impeding or killing the cancer stem cells. And that's why cancers come back, is because these stem cells eventually will repopulate the tumor. So is a lot of research right now where people are trying to find drugs that specifically destroy cancer stem cells, and that's certainly a very promising area right now. We'll, however, focus on the other three types. One other type of cell to talk about between stem cells and terminally differentiated cell, there's another type of cell called progenitor cells, mostly. And these are cells that have a very limited amount of replicative capacity, maybe 10, 15, 20 cell divisions. They can produce a few types of cells, not a great deal. But they are intermediate stages in that they are not terminally differentiated themselves. And if we see here, we have a stem cell doing an asymmetric division, and here we have a series of progenitor cells which are capable of dividing, multiplying, and giving rise to a large number of terminally differentiated cells. And these can be different types of terminally differentiated cells even. And these cells are also very important for lots of research and potentially medical uses. For instance, endothelial progenitor cells are types of cells which repair blood vessels and are therefore important in many, many types of diseases. Now, we remember from our talks about DNA replication and mutation rates that every time a cell divides, approximately 3 mutations arise in humans. And so therefore, if you have stem cells dividing over and over again, mutations might accumulate, and that could have disastrous consequences. However, it seems highly likely that, based on most of the evidence, that stem cells have a very unique ability to hang on to the template strand of DNA. Here you can see there's a cell division, and in normal cells dividing, it would be 50-50 chance about which cell gets the original one and which cell gets the copy. And after multiple rounds of replication, stem cells have the ability to hang on to the original template strand, always passing off the replicated strands to differentiated cells. So this is a way of diverting most mutations to cells that are not immortal, that are terminally differentiated, and the immortal stem cells sort of preserving a pure genome. But like almost everything in biology, you might not expect this to work absolutely 100% of the time. But nevertheless, it's doing much better than what you might expect statistically. So as you might be aware, the embryonic stem cells have been fairly controversial because many people consider the source of embryonic stem cells, the blastocyst, a fertilized egg at day 5, to represent human life, that the procurement of the inner cell mass revolves the destruction of an embryo. Therefore, people were encouraged strongly by law to not use a great deal number of stem cells or develop new stem cells for a while, and there was a lot of effort directed at using adult stem cells for the treatment of disease. And now iPS cells are also being explored intensively for these reasons. But there are advantages to each of the stem cell types, and we'll discuss them right now. Embryonic stem cells are flexible because they can form any type of tissue. We can have an endless supply because they can be proliferated indefinitely in vitro, so we can get large numbers. They're relatively easy to procure because they're early in development. They can have a lot of flexibility in their development. Some of the disadvantages, and which we'll explore in future videos, is that it's hard to get them to differentiate just the way you want it. Depending on the source, if it comes from a different embryo, they are non-self, and so the immune system will recognize them as foreign. They have the ability to form tumors, and again, a fertilized egg is destroyed in their procurement. Adult stem cells can also be relatively easy to isolate. Because they're already along the way to differentiation, we have somewhat greater success in getting them to turn into different types of tissues. Because you might be isolating them from you, they're not going to be immunogenic. They're less likely to be tumorigenic because they don't have the infinite replicative potential, and there's essentially no harm to the source material. Some of the disadvantages is that they are limited in quantity. We can't proliferate them in vitro the way we can embryonic stem cells. Eventually, they are going to stop dividing, and they may already exhibit signs of cellular aging, such as the accumulation of methylation and other epigenetic marks. So it's still a very open question in terms of what cells will end up proving most useful and easiest to use, but the immense potential of stem cell technology is why everyone is working so hard to develop it. Okay, thank you for listening.